In this module, we will discuss the phenomenon known as antibiotic resistance. So what happens is that when microorganisms, and especially bacteria, are exposed to the same drug, specifically an antibiotic, for a long time, then they develop a resistance through several mechanisms that we will cover in this module. So here I have a few examples of what happens and what is the disease, what's the agent, what was the drug initially active against it, and to which now they have become resistant. This is particularly important for the class of nosocomial infections that comprise Streptococcus aureus, several E. coli species, Klebsiella, and Pseudomonas. And those microorganisms are particularly dangerous because they are found in the hospital, so they reproduce and they grow in an environment that contains antibiotic or contains hosts that have been treated with antibiotics, therefore facilitating the emergence of resistance. This is particularly dangerous also because patients that are admitted to the hospital may have reduced ability to fight out infections, and so this becomes particularly important. And we've talked already on how vancomycin has become a particularly useful antibiotic to treat systemic infections leading to sepsis, but also that's an antibiotic to which resistance can emerge quite rapidly by simply changing the ala with the ala-DLAC. So let's talk a little bit about natural resistance and acquired resistance. So natural resistance happens when there is a particular gene at a chromosomic level that somehow contrasts the action of the drug. This affects almost all species strains, and this existed even before antibiotic was used, or at least used in the practice. And so, for example, bacterial species are resistant to amoxicillin. Acquired resistance, on the other hand, happened by mutation of the organisms, and so this can be found either the chromosomic, plasmidic, or transposon level. They affect the fraction of the strains. The fraction that has become exposed to antibiotic increases with antibiotic use. And for example, one of the things that happens with the E. coli is that they express a increased panel of beta-lactamases that are active against more antibiotic than just penicillins in E. coli. This is a well-known phenomenon. For example, if you remember the original experiment in which a penicillin was discovered, we found that in a agar plate covered with a culture of a strep, if one had basically the emergence of a mold, the mold will kill. So I'm kind of wiping out. Oops. The mold will kill all the bacteria surrounding it. Well, what happens after time, if we don't touch anything, is that we'll find that the situation changes completely, and slowly bacteria emerge that are resistant to the antibiotic secreted by the mold, and so they can grow in the presence of the mold. Okay, so what is the mechanism for resistance? We have several mechanisms possible. One is basically by preventing the antibiotic from reaching its target, and so that happens especially when an antibiotic needs to permeate inside the cell, so we can just change the membrane permeability, and we can also evolve transporters that facilitate the efflux of the drug. We can also prevent an antibiotic from binding its target, and so we can do that by expressing supplementary targets, and so overexpressing or producing more of the enzyme or the precursor that is targeted, or we can modify the target so that the activity decreases. And this is an example with the Diala D-LAC that we've seen for vancomycin. Another example that we've seen is the inactivation before reaching target, for example, what beta-lactamases do to beta-lactam antibiotics. So one important aspect of the emergence of antibiotic resistance, beside the widespread use of a certain antibiotic in the clinical practice, is the fact that animals are raised by adding antibiotics in slow doses to their feed. And so what happens is that this is done because it facilitates the growth of the animal and so the yield of meat. But at the same time, just like the unnecessary use of antibiotics in patients facilitate the emergence of resistance, what happens here is that we have typically very low doses of antibiotics, and those actually allow the selection of bacteria that are resistant to antibiotic, which then survive and multiply. That is spread to food directly through animal products, through produce that has been contaminated by water or soil containing fecal runoff. It can be done when food is prepared in an unsafe manner. So, for example, we have the same utensils used for dealing with meat and also for vegetables. And that's a problem because while meat is usually cooked and that takes care of most of the infections, then vegetables may not be cooked, and that's a source of infection. And also by the environment itself, when it's contaminated by fecal matter, coming from an animal where bacteria have evolved in a resistant manner. So how do the genetic modifications then spread out? There are two mechanisms. The one is that first we have the non-resistant bacteria, which then multiplies, and in the presence of the antibiotic, but also in the absence of it, we have random mutations in the DNA, and then in the presence of the drugs, only the resistant ones survive and take up the whole population. What happens then is that we have coexistence in the absence of antibiotic of resistant and non-resistant antibiotics. But then we have gene transfer between bacteria that are resistant to certain antibiotics and bacteria that are not. And by doing that, basically we also evolve a population of bacteria that is completely resistant. So let's have a quick case study with a nosocomial infection caused by Enterococcus, which is a microorganism that lives in the GI tract. And so what happens in this case is that while this microorganism is generally, pretty safe and not particularly dangerous, it can mutate in a manner that is out of control and basically become virulent. And we'll see exactly how that happens. This microorganism, as we say, it lives in the gut. However, it can move to the bloodstream either by going through the portal vein to the liver and then take up particularly in the heart valves, causing very dangerous endocarditis. Or it can be contaminated in the environment through the GI tract, basically through contamination of the skin of the patient surrounding the perineus, or also contaminated in the environment if the hygiene is not kept very well. So what are the mechanisms that cause this otherwise pretty mild microorganism to become virulent? So what we have is that in the absence of antibiotics, The intestinal lumen contains a variety of gram-positive and gram-negative organisms. And here we have the gram-negative in pink and the gram-positive in purple. And those endosomes are also kept in track by secretion of this lectin called reg3 gamma, which binds to bacteria and again facilitates the involvement of the immune system to prevent any population from taking over. When we have antibiotics, we have that the gram-negative organism decrease. Also, we have the decrease in secretion of this lectin, and so the gram-positive bacteria take over and multiply in a manner that is not regulated. And because we also have the decrease in this lectin, we cannot really prevent the spread of these infections. And those are the situations in which the Enterococcus can take over the population in GI tract and become virulent. Virulent simply means, in this case, that it can multiply in a manner out of control. Okay, so with that, I also want to give you an overview of various antibiotics and what are the mechanisms for resistance that can be expected. So for aminoglycosides such as gentamicin and streptomycin, we can have two levels, two types of resistance. One is simply by decreasing the uptake of the antibiotics. The other one that is much more important and much scarier is that we have enzymes that modify the antibiotic so that it's not active anymore and cannot bind to the ribosome target anymore. We have already talked at length of mechanisms by which beta-lactams are not, basically a resistance emerges to them, and besides the beta-lactamases, we simply have an increase in penicillin binding proteins that basically take up the penicillin and reduce the binding affinity to the target. We have seen for vancomycin the mutation of diala to diala-dilac. We have also seen in other antibiotics simply the alteration of the interaction with the cell membrane, and that's true for lipopeptides. And in general, those are the main mechanisms that we'll find. We also have some mutations in the small subunit of tRNA or in other modifications, but also those tend to be more rare because the ribosome, of course, is very highly conserved. So with that, we had an overview of bacterial resistance and the mechanisms that give rise to it.